Okay, so neurons. Neurons are, the cells that we often focus on in a course on neuroscience, right? And they just come in an extraordinary variety of shapes and sizes. You have extremely small, what are called granule cells in the cortex and in the cerebellar cortex, and you've also got extremely long cells. Some of the largest cells, actually the largest cells in the body, that are actually these touch sensory neurons that you find in your toe, where if you press on them, they transduce or convert that pressure into an actual electrical signal that'll run up this wire-like extent all the way into your spinal cord, and then up to make their first synaptic connection on neurons that are located in the brain stem. So an extraordinary variety of sensory neurons. These are called afferent neurons that are transducing various forms of energy out here that are impinging upon you and within you as well into the language, electrochemical language of neuronal transmission. And also efferent, which are neurons that are sending information from the central nervous system out, basically to move the body or influence aspects of bodily function. A lot of interneuronal cells that are found within between the afferent input, the sensory input, and the motor or efferent output. So an extraordinary variety of shapes and sizes and functions. Neurons are really rather remarkable. 86 billion of these cells, in the average adult human brain. But there are often, a number of discrete kind of both structural and functional compartments that characterize neurons. And I'm going to use this as like a sort of classical neuron that I composed out of pipe cleaners. And it has a number of discrete sort of functional regions that carry out specific, aspects of neuron function. So, for example, you see all these incredible branch-like extensions at this end here, right? These are called the dendrites, which is the Greek word for branch or branches. And these are membrane bound. So they're like, branch-like projections, but they're all surrounded by phospholipid bilayer with, various protein machines embedded in those membranes, including receptors and channels and, transporters and everything else. The dendrites allow for a greatly expanded surface area for connections, from other neurons. So neurons will come and make synaptic connections, up to 10,000 distinct synaptic connections, on a single neuron, depending on the number of those dendritic branches that you actually see. Actually, some really fascinating work that was done by Marion Diamond, a researcher in neuroscience, examined the growth of, additional, portions of dendrites, these little, spine-like kind of bulges that came out all over in response to enriched environments. So in enriched environments with lots of activities and lots of stimuli and lots of things going on, you actually see further elaboration of dendrites, the growth of these dendritic spines that allow for more surface area and more points for synaptic connection. But the dendrites are really the sort of input end of a neuron. This is largely where excitatory inputs come. We're going to see these are, the release of a neurotransmitter that'll be, received by a receptor here on the dendrite. Receptors that can, push the neuron towards firing and sending an electrical message down its length to its ends, or an inhibitory, input. It'll be the release of a neurotransmitter from another neuron, that'll be received by, other receptor proteins that will, shift the conditions here that'll make it less likely for this particular cell to fire. So think of up to 10,000 distinct synaptic connections all wrapped with their astrocytic membranes, basically, being received, all these messages being received at the dendritic end. So the dendrites are sort of the input end, basically, of the neuron. And then hidden within all this forest of dendrites here, of branches, is a spherical cell body of the neuron. This is where the nucleus is located. Another word for the cell body is the soma, which is Latin for body. And the nucleus here, of course, contains the genetic material, the chromosomes, the DNA that can be transcribed, and then translated, into the protein machines, including all of these, various receptor proteins and channel proteins and other things that we were describing and explaining earlier. So basically, the cell body is essential for sort of cellular upkeep and cell maintenance, right? The synthesis, the expression of genes, the synthesis of proteins, etc. And then at the very beginning of this wire-like extension here, this wire-like extension is called the axon, is a bulge. It's actually like a little hill, it's described as. It's called the axon hillock. And it is just packed with various protein machinery, including a lot of voltage-gated channels that we'll talk about that are involved in the decision about whether to initiate and, send a current, down this wire-like axon, to its end. So the axon hillock, we're going to see, is a very important, structural component of a neuron, which is just loaded with, protein machines that can, make a decision. If the environment at that hillock, hits a certain, setting, well, then you're going to get the initiation, of a current or electrical message away from the cell body, down the axon to its end. So the axon then, which could be very short or, in the case of other neurons, very long, is the wire-like extension that sort of sends electrical messages away from the cell body and carries information in the form of current, currents that are going to be going back and forth across the membrane, we'll see, and be propagating, working their way down from one end of the axon to the other. And at the very end, we have, first of all, you can see the axon can branch. It forms what are called collaterals. But ultimately, these currents are going to reach the very tips here. And there's going to be a conversion process from the language of electric current, from the flow of that electric signal down this, wire-like axon to the release of a chemical that's called a neurotransmitter. So the axon carries the message away, and the axon terminal is the site of conversion of the arrival of the electrical message into the release of a chemical message, and it'll form a synapse, with another neuron, with another cell. So these can form, ultimately these networks of linked neurons. So again, dendrites, input end, soma, cellular upkeep, cell body here, nucleus, protein synthesis, gene expression. The axon hillock is the decision-making point on a neuron for whether it's going to fire or not. The axon carries that, information, down its length in the form of an electric current. And then the axon terminals is, at the very end here, this is where the release of neurotransmitter, the conversion of the electrical signal into the release of neurotransmitter will actually occur at synapses.